Hey folks, and welcome back to another edition of Zog Science. Today we are going to be doing our first asynchronous lesson for AP Biology, and we're going to be taking a look at data analysis. So why do we need to do data analysis? Well, whenever we do some sort of experiment, we collect data, but we don't, know what that data is actually telling us. So we need to analyze the data in order to help us to be able to interpret it and to give that data meaning. Data by itself doesn't really tell us anything. We have to actually interpret the data. This is often called statistics. So there are two main types of statistics, descriptive and inferential, and we're going to talk about both of those today. In descriptive statistics, just like the name implies, we are figuring out things that are, or just calculating things that are going to help us to describe a data set. So examples, and the ones we're going to be learning about in this class are mean, standard deviation, and standard error. There are lots of other types of descriptive statistics as well, but these are the ones that we're going to be talking about here. And then in inferential statistics, we're going to be comparing two different data sets, and examples would be t-test and chi-square. So when we talk about the mean, right, I think that we all have a general idea of what that means, right? That's the average. It kind of describes the middle of the data. Data is often thought to be kind of normally distributed, which means that you have a curve that kind of shows out, and I'm going to show you what that looks like in just a moment. And so the mean is kind of the middle of that. And it takes into account the natural variation that occurs when you're collecting data. If you think about like the heights of, a population, you're going to have some people that are really tall, some people that are really short, and then you're going to have a lot of people that are kind of in the middle, or a lot of people that are really close to the mean. The more data points you have, the more accurate the mean. And so if you take a look here, this shows you the formula for calculating the mean. It's a lot of complicated things. Basically, here's your symbol for mean, and that's going to be equal to the sum of all of your values divided by however many values there are, right? So just your normal, what you do when you find the average, you add up everything and then divide it by the number of values that there are. This, by the way, is sigma, and this means sum. Our second value is standard deviation, and this is a way to describe data and how spread out it is from the mean. And so the more spread out, the higher the deviation, or the standard deviation, which essentially means that there's more outliers. And it can indicate that your experiment wasn't very well run, right? Because if you think about it, if you end up with really spread out data, that could mean that you didn't have an experiment that was set up very well in terms of kind of controlling for a lot of factors, so you ended up with a lot of variation. It doesn't necessarily always mean that, but that's what it can mean. If we take a look at this picture, here is our mean. That's what the vertical line is representing. And then this is what we were talking about, that normal distribution. Notice how there is an equal portion of data that is both below and above the mean, and it's normally distributed. It's on both sides. The one with the lower peak, this represents a high standard deviation. Notice that the data is much more spread out versus the low standard deviation where the data is much more centered around the mean. Here is our formula for standard deviation. You're going to sum up the difference between each of the values subtracted by the average. So like, let's say you had 10 values. Well, you would have to do that 10 times. You would take each value and subtract the mean. You square it, divide by n minus 1. So again, if we had 10 values, we would be dividing by 9, and then we would square root that number. And then finally, our last descriptive statistic is standard error, and it's technically standard error of the mean. And standard error tells us how far the mean from the sample is from the actual population mean. So when you do an experiment, right, you're collecting data on just a sample. You're not actually collecting data on the entire population. Okay, standard error basically tells us how far away is the sample mean, right, the mean that you calculated, from the actual population mean. And a larger standard error indicates that the actual mean can basically fall into a big range. A small standard error would mean that the actual mean is very close to what you found in your sample. And standard error bars often show up on graphs as whiskers. So for example, here, what we're showing here, this is the mean is at 1. However, with the standard error bars moving both above and below that by it looks like around 0.25 or so, what that means is that this is the sample mean, but the actual population mean could fall anywhere between those bars. Here we have a much smaller standard error, and then this would be a much larger one. Now, if you were to take a look at this graph without these whisker bars, you would say, okay, there's definitely a difference between these three different treatments, right? You can easily see that with the means. However, the standard error bars show that actually there's quite a lot of overlap between these, right? If we look at the standard error bars here, this overlaps with most of the standard error bar here, and the uric acid, again, it overlaps a lot with the other ones as well. So it is possible that the actual population means could be all the same, right? The population means for all three of these different treatments could all be the same. And so this is a way to kind of be able to interpret your data and say, okay, is there an actual difference between my different experimental groups, my different treatment groups here, or are they all the same? And here it is certainly possible that they could all be the same because of the overlap. Standard error is calculated by taking the standard deviation and then dividing it by the square root of n. So it's derived from the standard deviation. So, if you see a smaller number here, that likely means that the standard deviation was also smaller for the oxalate versus the uric acid probably had a much larger standard deviation. Now, this does depend upon the sample size. So if you have different sample sizes, that's something to take into consideration as well. So that is descriptive statistics. Let's talk about inferential statistics a little bit more. If we have two sets of data, so again, let's think about our phosphate versus our oxalate. We can see that the standard error bars overlap a little bit, but the means do really look different, right? How do we know that those means are actually statistically significantly different? Well, we wouldn't be able to know that just by looking at them, right? Even if the means look a certain distance apart, we can't tell that just by looking at the data. So inferential statistics are used to answer that question to determine whether or not it's actually significantly different from each other, and so therefore they're sometimes called significance tests. To start with, you're going to need to write a null hypothesis, and that is what you're actually testing. So when you came up with your hypothesis at the beginning of the experiment, that was for your overall big experiment. The null hypothesis is specifically for the statistical test, and it is only something that can be answered using the statistical test. When you write a null hypothesis, you're going to write it such that you assume that the data sets are the same. And we want to assume that they're the same, and then you have to prove that those data sets are different. So for example, there's no significant or statistical difference between the number of hamburgers sold between Chapel Hill and Carrboro, right? That would be a null hypothesis that then you would run your statistical test to determine whether or not that is true. So the first test that we're going to talk about is a t-test. A t-test is a very accessible test that can be used in a lot of different ways, and it is a test that is not going to show up on the AP exam. However, there are a lot of data sets that you can only use a t-test for, and it's basically used to compare the means of two sets of data. So when we do a t-test or any sort of statistical test, we want to use a p-value of 0.05, and the p-value is basically our confidence in our statistical test. So a p-value of 0.05, if we are able to pass the test at that level, that means that we are 95% confident that the difference in the means that we see is due to whatever we did in the experiment versus it just being random variation, right? Because again, two samples, you're going to have random variation. So when you run the t-test, it's going to give you a p-value, and you have to compare the p-value that the test gives you to that p-value of 0.05. If the p-value is less than 0.05, then we reject the null hypothesis. But remember, our null hypothesis was written in the negative, right? Stating that there is no significant difference. So when you reject the null hypothesis, you're saying that there is a significant difference. If the p-value is greater than 0.05, then we fail to reject the null hypothesis. And therefore we are saying that we can't tell, or our data doesn't have enough evidence to show that there is not a significant or statistical difference between the number of hamburgers sold. It is possible that the difference that we see in the mean is due to our experimental condition, whether we're measuring hamburgers in Chapel Hill or Carborough, but we don't have enough evidence to actually support that conclusion. It could just be due to random error. We're not confident enough. So again, when you reject the null hypothesis, that means that there is a significant difference between the sets of data. So normally when we would do this, we would be in class and I would have, you all get out your graphing calculators, and I've got some in class that you could borrow, and we would be doing this using graphing calculators. However, I recognize that not everybody has graphing calculators at home, and so I'm going to show you all how to calculate these statistics using Google Sheets. And basically what you're going to need to be able to do is to use the formula functions in Google Sheets, or I think in Google they're called functions. Maybe it's in Excel they're called formulas. But regardless, when you're doing this, you're going to basically put your data into columns, and then you're going to be using the equals sign to then write each of the functions. I'm going to show you what that looks like in just a moment. For the mean, you use the average function. For standard deviation, you use the equals stdev, or standard deviation formula. For standard error, you have to write a custom formula. There is no standard error built into Google Sheets. You'll just have to write your own formula. But remember that standard error is just the standard deviation divided by the square root of however many items you had. So it's a fairly simple formula to be able to input. And then for the t-test, you can use the equals t-test function. So let's take a look at our example, and I'll have this linked for you all in the assignment, so you could open up this doc yourself. And if you want to save a copy to reference, you can always do that. So in our first example here, we have trichomes, and trichomes are little hairs that grow on the sides of plants. They're an evolutionary trait that basically helps kind of keep off insects and other herbivores from eating the plant's leaves. So here's my two sets of data. And you'll notice that what I've done is I've typed in mean, and let me open this up a little bit, mean, standard deviation, standard error here. And then what I've done is I've typed in my formula. Now I'm going to get rid of these over here real quick to show you how to do this. You'll notice that I've typed in equals average, and then I've done those, and then I've selected the range here. So I'll delete this and show you how to do that. So you type in equals, and then average, and you'll notice that Google Sheets is smart enough to start pulling it up, and it will actually autofill all of the data sets that is above that. So it will actually go ahead and suggest that and pull it in. Now let's say that you didn't like what the range that it was suggesting, or it wasn't pulling it up. What you would do is you type equals average, you will then open up the parentheses, and then you just click and drag for the data set that you want, and then close the parentheses and hit enter, and that will do just auto calculate for you. Same thing with standard deviation, equals standard dev, and then you have your data range, and then that'll give you your standard deviation. Now for the standard error, two different ways you can do this. Number one is you can use the formula that I've got here. I'm using the count a function. The count function basically just calculates the number of items that are highlighted here. So basically it's just telling me what my n value is. Or what you can do is you can manually type that number in. It's however you want to do it. You do have to use the square root function, and you got to be careful with your parentheses. So notice that I'm opening up 3 sets of parentheses here, which means I have to close 3 sets of parentheses here. And notice also that I'm using the cell function here in order to select that cell. Now, one of the nice things about Sheets and Excel is that let's say that I've got this typed in for one of my data sets, but I don't have it for the next data set. Well, rather than having to type all of those, what I can do is I can highlight, and then this little box right here, I can click and drag, and it will copy over the formulas, and notice that it will also change my B, right, column to my C column, and it will just go ahead and do all of that. And now I've got my 2 sets of data that I can compare. Now for the t-test, it's very similar. You just type in equals t-test. You then need to do your 2 sets of data, and then there are some different things for this one. So if I click on the t-test in the formula, notice that it says range 1, range 2. That's my 2 sets of data. Tails, so basically the tails are the 2 sets of distribution. We'll almost always be doing a double distribution, so you want to put 2 there. And then the type. The type is basically the type of t-test. There are 2 main types of t-tests. There is a paired t-test, and then there is an unpaired t-test. And so for your paired t-test, what you want to do is you would select 1 for this final number. A paired t-test is where, let's say that I have a set of individuals, and I measure what they can do, let's say reaction time before a treatment, and then a reaction time after a treatment. That would be a paired test because it's the same individual before and after. These are different plants before and after. I'm just measuring that there were 10 plants before, or not 10, but 17 plants before, 17 plants after. And so that is, this would not be a paired test. And in that case, you would want to select 2 for that final type of test, okay? Notice that, again, it spits out a p-value here. This p-value is greater than 0.05, and so we would fail to reject the null hypothesis. And so even though my means here are different, they're not different enough statistically to be able to say that the differences is caused by whatever we had before and whatever we had after. If we take a look at example 1, it's all set up exactly the same, but notice again that this time my t-test comes out, it is different. I'm sorry, my t-test comes out and my p-value is very, very low. My means are a lot different this time, and so therefore, because my p-value is less than 0.05, we are going to reject the null hypothesis. All right, so that is all for today in terms of the data analysis. Make sure that you know how to work these things and how to use them. One thing that you can also do, all of these formulas that we are going to be using, if you search for AP Bio formulas, the formula sheet, you will see the formula sheet that you would actually get to use on the AP Bio exam. You'll notice that it has the formulas for the mean, standard deviation, standard error of the mean, and then it also has this other test, which is the chi-square test. We're going to learn about that a little bit later in the year. That is another inferential statistic that is useful in different situations than the t-test, and again, we'll come back to that at a later time. So, thanks for watching, and I will see you all soon.